if you're interested, we can always chat after class. I'm always about the learning. All right, molecular biology. It's game time. Game time. Central dogma. Woo! Who's excited? Okay. So we begin our story here with a couple very famous scientists and DNA. Now, does anyone know the name of any of these scientists? Not, that's okay. That's why I'm here. And maybe you have heard these names before, very famous. So this is Rosalind Franklin. So Rosalind Franklin was working as a biochemist, molecular biologist, trying to discover the structure of DNA, what it looked like. And so she was working using a technique called X-ray crystallography, which we still use today, more so for the structure of proteins we're interested in. And her very famous image from her lab is also on this slide here, using X-rays to determine the structure of DNA. And this is a top-down view. So looking up, down, downward, downward? Sure. Above DNA, looking down. And that was enough. So these two guys, Watson and Crick, just kidding, Crick. I say that because, as I think Patricia just messaged, there's this kind of, it's not really a myth so much as it a controversy, right? That they stole her work, she didn't get the proper credit. And I think there's certainly an element of that. There's no doubt. But essentially, Watson and Crick, these two scientists here, were in the neighboring lab. They saw her data here, this image, and they were able to deduce the exact molecular structure of DNA pictured here, which we're going to talk about. And so those, oops, what did I just do? Sorry. Wrong button. Sorry. Okay, so in 1953, it's about the only history we talk about, and you don't have to know that date, but they won the Nobel Prize, Watson and Crick, for determining the structure of DNA. And what they saw or figured out is it, as you saw on the slide, it's this double helix. The DNA is a double-stranded helix. So a helix is like a spinning kind of ladder. Essentially, it's a ladder that's spinning around with these helices. So structure of DNA as double helix. And again, these two scientists are Francis Crick and James Watson. Still alive today, I think. It's been a while. But they would give talks and stuff at Harvard and stuff. They're very famous. And so again, DNA is a double-stranded nucleic acid. So DNA is a double-stranded so remember from our very first unit, talking about the different types of biological molecules, it's a nucleic acid, right? Nucleic acids, proteins, carbohydrates, lipids. So nucleic acid was one of those categories of biological molecules. And we can also say it's a double helix. So let's look at that. Okay, so that image on the right here, that is the structure of DNA. We're going to take a little more of a detailed look. Okay, so calling back to Unit 1, DNA was a polymer. It's an example of a polymer nucleic acid. And the monomer unit, the repeating unit that is linked together by the covalent bonds, is called the nucleotide. If you remember the nucleotide. So we'll briefly review the structure of a nucleotide because it's very important to the overall structure of DNA, which is our genetic material. And we just finished genetics, right? And I just said that DNA is our genetic material, but really it wasn't until 1953 and molecular biology techniques advanced that we understood that DNA was the genetic material. Mendel was, a long time before it. He didn't know it was DNA. He called them heritable units. He didn't know it was DNA, chromosomes. But anyways, I digress. So let's take a look at a nucleotide again. Consists of 3 parts, right? As shown here. So nucleotide are monomer, composed of 3 parts. And I'm leaving space over here because I'm going to draw DNA, draw it out. So the first part, starting on the left of that picture, is the phosphate group. Phosphate group is joined to a 5-carbon sugar molecule. And then the third component of a nucleotide is our nitrogenous base. Of which we have 4 varieties in DNA. So now I'm going to sort of talk about specifically DNA and the nucleotides of DNA. Because RNA's nucleotide, it's a different polymer, nucleic acid. RNA, their nucleotides are slightly different. Not drastically, same basic thing going on, but slight difference. And we'll discuss this. But in DNA, does anyone know what DNA stands for? Without Googling it? Ha ha ha. Let's use red? No. Do I have any other colors? Let me see. No other colors. Let's use orange. Nah, it might be hard to see. Brown. DNA for brown. Deoxyribonucleic acid. Deoxyribo. It's all one word. So DNA equals deoxyribonucleic acid. DNA. Okay. Why did I tell you that? Well, it's named that way for a specific reason. Deoxyribo. That's referring to the 5-carbon sugar. The 5-carbon sugar in DNA nucleotides of DNA, the nucleotides that make up the DNA polymer, they all are deoxyribose sugars. That's the name. And I'll show you why, or why that is. So deoxyribose is the name of the 5-carbon sugar in the nucleotides of DNA. Let me show you why. Notice this group here, or this carbon. So the carbons aren't drawn in organic chemistry. They leave them out. But I want you to see that off of this second carbon, number 2 here, which we call 2', 2' carbon. Sticking off, this hydrogen doesn't have an oxygen attached to it, does it? In contrast to the third. So that's why it's deoxy without oxygen. So it's a deoxyribose sugar. Okay. So that's one unique thing to DNA. Okay. What else? What else is unique to DNA? The nitrogenous bases. So something that's not unique. There's 4 varieties, right? There's adenine, cytosine, actually. Let me just write thymine here. Never done this, but I think this will help. Thymine is unique to DNA. The nitrogenous base thymine. So when we talk about RNA and other nucleic acid polymers, which really there's only RNA, it doesn't have thymine. It's unique to DNA. And then the other options are guanine and cytosine. Sort of ran out of room, but guanine and cytosine are the last 2 possibilities. Cytosine. So we have A's, T's, G's, and C's. One more thing I want to mention, because it's important to DNA's overall properties. Does anyone remember something about the phosphate groups? We've talked about them a couple times now. Well, what's characteristic of these phosphates? can show you the picture again. What do you notice about that phosphate group? Not electronegative, but it's a good close answer for sure. But just the fact that it's negatively charged. So minus charge. So DNA, and this actually is something in your lab as well. It explains something that happens. But DNA has an overall negative charge to it because all the nucleotides have these highly negative phosphate groups. Okay, so what does DNA look like if we zoomed in? Let's draw an example of one of these, or some of these nucleotides strung together in a molecule of DNA. I guess I'm sticking with brown for DNA, so let's do brown, I guess. All right. So here's how I'm going to do it. So I'm going to start with a phosphate sticking up. Phosphate linked to a sugar, so I'm just drawing one nucleotide right now. And then I can choose any of the 4 bases, but I'll just do A, adenine. Okay, well, this sugar is going to be connected to another nucleotide, phosphate, sugar. And then for this one, I'll put cytosine, C. Okay, cool. We'll just do one more, phosphate, sugar, and I'll do a T, thymine. So that's one strand of DNA, right? We know that DNA is double-stranded. So let's draw the other strand. And we actually start here with a sugar. Phosphate is going to come down. Here's the base. Kind of want to leave a little space here because I got to show you something, but this is going to connect to a T here. So this has to do with complementary base pairs. So the way the chemical groups are arranged of these nitrogenous bases, A's always pair with T's, always. A's and T's. C's only pair with G's. So this we would call an AT base pair. AT base pair. This we would call a CG base pair. Okay, and then I drew one more, right? So we got our phosphate, sugar, and then another phosphate sticking down. I'll explain that in a minute. But the sugar is where this, so what base would pair with the T here? Yeah, adenine. Okay, that's the general structure of DNA. Seriously. Now, there's a couple little extra details you have to know about the way DNA is arranged like this. You'll notice I kind of drew it upside down to one another, like this. Like this one has a phosphate sticking up, but this one has a phosphate sticking down. Well, in reality, DNA strands run anti-parallel to one another. That's what we call this. So it's the strands run opposite directions of one another. So it's like you flipped it upside down on the other side. So it's the anti-parallel nature of DNA. Okay. The other thing I want to mention is due to this anti-parallel nature, one strand here, this strand, is going to end with a certain carbon sticking up from this nucleotide. It's going to be the 5th carbon. And I'll show you a detailed picture of this. I hope I have it in my slides. If not, well, I'll take a look. But this end with the phosphate sticking up, this is the 5' end. So again, this is 5'. The prime is only because there are other carbons attached to this sugar, right? In the base, in the phosphate, actually not, actually in just the base. So they just started over kind of thing, but you don't have to know that. You just have to know that there's a 5' end, and then this end will have an OH group sticking off. So the O covalently bound. So let me write the H over here, actually. So the OH sticks down. And this is our 3' end. So the 3' carbon sticks down. So this is our 3' end. And like I just mentioned, this anti-parallel nature, because it's upside down, this would be our 3' end up here on this strand. And this would be our 5' end. So why am I telling you this? Well, I'll show you. Well, essentially, when you read a DNA sequence, it'll say 5', 3' on this side, and then 3 over here, 5 over there. And it's an important concept because when we talk about how DNA is replicated and how these enzymes work, they only work in certain directions. So it is important to know this. Okay, one last thing I want to say about this structure here. How are these attached? Again, we're going to do a callback to Unit 1. I love how biology is so interconnected. It's not like we learn something and then forget about it. It's just not important anymore. No, hydrogen bonds. So, thought you'd never hear that again, did you? The bases, the nitrogenous bases in our double-stranded molecule are actually connected to each other by hydrogen bonding. Oh, what was that? Is everyone muted? Oh, I forgot to mute everyone. There we go. Okay. So let me check notes here. Okay. A's and T's, again, because how the molecules look and facing each other, will have 2 hydrogen bonds between them. So 2 hydrogen bonds. C's and G's, on the other hand, have 3. So they're a little more stronger bound. 3 H bonds, hydrogen bonds, between C and G base pairs. So again, this would be 2 here, right? And again, I like to make everything important and relevant. I don't want to just teach you things for no reason. The fact that there's hydrogen bonds here is really nice, because if you go on in biology, you'll learn that we're really unraveling our DNA a lot. We need enzymes to come in to replicate DNA. We need things to come in to do what we're going to talk about next, the central dogma. We've got to take it apart a lot. This is a great system because hydrogen bonds, we learned, are very weak. They're weak intermolecular bonds. And so the pulling apart of the DNA isn't this energy expending, like, crazy thing to pull it apart. It's, loosely bound. Granted, because there's billions of these base pairs, it's pretty tight. But if you want to go to one little region, then you can just access it pretty easily. It's pretty good. So there you go, making chemistry relevant, right? We learned chemistry, didn't think it would apply to biology, and here we go. Here we are. Okay. So let me see on my slides if I have this picture I want to show you. I feel like I added it. I hope I did. Damn it. I mean, darn it. I'm going to find it. I'll find it on Google. It's really quick, and then I have to add it. I didn't I had my, you guys can see that, or we're seeing this, right? I wasn't sharing the whole time. Was I? Did I forget to hit stop on the share or no? Okay. Okay. Sometimes I forget. Or not forget, but I'm so used to Zoom now. I just kind of, my brain spaces out, I guess. I'm kind of, I want to teach in person again. Okay, let's find this cool image. DNA structure. Ah, here it is. Okay, so check it out. Just on the right side, that's the more complicated version, right? You can see the hydrogen bonding between A's, T's, C's, and G's. But really, I wanted to show you as well this 5' 3' thing, how the 2 strands are oriented upside down to one another. And so if you take a look, I like this picture because you can see the carbons, right? And you'll see that the 5' here is sticking up next to the phosphate in the sugar molecule. And then on this side of this strand, the other side, you have a 3' with a OH or hydroxyl group sticking down, the 3' carbon. So it runs 5 to 3. The other side is flipped, right? Everything's upside down to one another, how the molecule looks. And so the other strand runs 5 down here, 3 up here. So you'll always see it like this, anti-parallel, not parallel. Okay. Sweet. Any questions before we go on to DNA replication?
Are you going to get through all of this?
The last thing we covered
only
today.
have like 10 minutes. So are you going to be able to get through all of it?
What's that? Yeah.
Okay.
Well, we have another lecture for molecular bio, so we'll finish it next time if you know.
Can you give us a link for that picture?
Oh yeah. I'll email it to you guys.
Thank you.
You got it. Yeah, I thought I had it in my slides. I like that picture, so I'm going to add it. Email DNA pic. Okay. So DNA replication. All right. So we've talked about DNA replication. It occurs, who remembers when it occurs? Prior to cell division, right? So what phase did it happen in? When does DNA replication happen? Probably trying to forget the last unit. I don't blame you. It was a lot. Okay, no worries. S phase of interphase, right? DNA synthesis phase. So before mitosis or meiosis, DNA is replicated. So we're going to talk about how that is, how that happens, sort of. Not too much detail. We'll leave that to the majors courses. But the general concept here, we have our DNA double helix. And what happens is the DNA strands that are connected by these hydrogen bond base pairs will separate with the help of some enzymes. So here it is separated out. And a very important enzyme comes in now to replicate these DNA strands. make copies? Well, complementary base pairs. So what does that mean? This enzyme is going to come in and add complementary base pair nucleotides to these two strands. So if there's an A here, it's going to come in and put a T nucleotide. G here, C. T here, A, etc. And so you end up with two identical to the original parent DNA molecule. This enzyme is called DNA polymerase. Note how it's DNA polymerase. There are other polymerases, but this one's DNA polymerase. Star it, because it's an important one for this. And this is the enzyme that copies DNA into a complementary strand of DNA. So I'm going to write synthesizes complementary strands of DNA. Complementary strands of DNA. Okay, one last thing here before we wrap up the lecture. This mode of DNA replication took a while for scientists to figure out how it happened, but we call it semi-conservative DNA replication. Notice how in these two new DNA molecules, half of it is from the original molecule. These are called template strands, templates to the new complementary strand. So this mode of DNA replication is semi-conservative. Half of it is conserved, it's kept from the original molecule. So this is semi-conservative DNA replication. Where half of the original DNA is kept as the template strand for the new complementary DNA strand. Complementary DNA strand. You can just write strand because I ran out of room, or I'll just add DNA. There you go. So we call this semi-conservative. All right. Sweet. That was perfect because now we can devote all of next time to the central dogma. Now that we know the structure of DNA, now that you know how DNA is copied or replicated. So any questions about today's lecture? And then I can talk about the lab or whatever, any other questions. Oh, let me unmute.
Okay, I just want to verify that when you're doing these replications, that they are matching the A with the T and the C with the G in that picture?
Yeah.
Okay.
So if I, in my original, and we'll practice this next time too. But if my original template strand here is like, let's say G, A, C, T, A, then DNA polymerase is going to come in and add complementary nucleotides, which would be here C with a C base, T, G, C, T. Oh wait, A. Ha! I did a mutation. A to T, right? Yeah. Is there another question? For the post lab.
Yeah, how long are they?
Oh, how long are DNA strands?
Yes.
Much longer than this. Much longer than 5. So genes can range from a few hundred to a few thousand nucleotides. But amazingly, our genome, the entirety of our DNA, is roughly 3.2 billion base pairs long. So when our cells divide, all 3.2 billion have to be copied and be correct. And it does this really well and pretty efficient. So it's pretty amazing. And yeah, so it's like really fast. Okay, Deborah, I got a message for the post-lab question 1. Okay, first let me see if there are any other lecture questions, and then I can answer lab questions. Okay, well, great. So that's it, but yeah, lab. So Deborah, for the post-lab question 1, could you explain what it's asking us to do? Yes, absolutely. So let's talk about the lab. Okay. Gonna sit down, get some water here. And I have the lab over here. Here we go. All right. By the way, if you're ever wondering about the sunscreen and I was swimming, and a tank top is, gotta shut the window, it's too hot. So sorry if I don't look professional. Post lab question number 1 from